All right, thanks so much, John. It's great to be here and telling you about our microbiome work. So as John mentioned, I'm part of Berkeley Lab. We're part of the biosciences area at Berkeley Lab. We have one of the biggest biosciences research programs of the national labs. And I'm actually affiliated with two different parts, the Joint Genome Institute, which is where Zhang is from and where I've spent most of my time at Berkeley Lab, which is a user facility providing genomic capabilities to the research community, specifically focused on bioenergy and nutrient cycling. And I now lead a division called Environmental Genomics and Systems Biology that has a similar science mission, but is more grant-driven research. But so a lot of the work I'm going to talk about today was done at JGI, but also in the AJSB. So just quickly, I'm going to talk a little bit, since this is a microbiome session, you guys probably already know a lot about microbiomes, but I'll give a little background and then talk about mostly our work on the San Francisco Bay Delta and carbon cycling and wetlands there, as well as a little bit about some of the future work we hope to do. So microbiomes, I love to talk about microbiomes. So even though it's a microbiome audience, I just remind everyone that microbes are ubiquitous. They're everywhere. They're in all kinds of extreme environments, not just on humans or on plants, but in these hot springs and below the ocean and all kinds of places. But they're kind of hard to study with the conventional methods that people have used for a long time in microbiology, specifically cultivation in the lab. And historically, it was found that, even though you knew there were microbes in environment based on microscopy, et cetera, when you plated those out on an agar plate, you tended to get a lot less colonies. And they actually called this the Great Plate Count Anomaly. And people talked about, well, maybe they're just not in a growing state. Maybe there's, various reasons. But the reason that it turns out to be is that most of these organisms just really don't like growing in the lab. But then the ones that grow in the lab are actually really different from the ones that are out there in the environment. And most of that has come about because of technology, especially sequencing technology for RNA and DNA. And I like to give this primer because often people don't really know much about 16S ribosomal RNA. This is just an image of the physical structure of the ribosomal RNA, which is unique compared to coding RNAs in that it has this structural base pairing that makes it really highly conserved. So the ribosome is, of course, central for protein synthesis, essential to cellular life. And these structural RNAs, therefore, if any mutation disrupts that base pairing, it can disrupt the fundamental function of protein translation. And so it is very well conserved across all cellular life. But also in these non-base paired regions, there's a little more flexibility. And so it actually turns out through the molecule, there are parts that are very highly conserved, and then there are parts that are more variable. And that makes it perfect for this barcoding use that we use it for, which is that we can amplify based on those highly conserved regions and then sequence through the variable regions and use that as just sort of a marker for a particular organism. And the first person to actually propose that as a technique for understanding molecular phylogeny was Carl Woese. And this was just an amazing change in microbiology, which really wasn't that long ago. This is a paper from 1987. Before that, no one really knew how microbes were related to each other because they had to rely on things like shape or metabolism, to try to build a tree. And being able to build a molecular tree based on real sequence information just completely changed people's understanding of microbes and how they were related to each other. And then the next step from that was applying this to those uncultivated organisms out in the environment. And Norm Pace had this idea that there were these samples where they couldn't seem to grow the organisms in them. They could take things from this hot spring at Yellowstone back to the lab and nothing would grow. And then he said, hey, but we can actually pick this up in our hands. Why can't we just do these sequence-based characterization on the samples from the environment? And they did. And I was talking to Phil Hugenholtz, who's involved in this work. He was like, it was like discovering there was this whole wide universe out there that we had no idea was there, because the things that came out of that analysis were just totally unlike the sequenced and characterized microbes that people have been growing in the lab. And so that led to this huge expansion of the phylogenetic tree, not just for bacteria, but for archaea. And to the point that really there are more uncultured lineages than ones with cultivated representatives. And that's continued to be the case in microbiology broadly. And it kind of gave us this picture that what we're looking at with those cultivated microbes is just kind of that tip of the iceberg. And there's this huge diversity out there. And that the next natural conclusion is, no matter really what you're looking at in the environment, which is what we really are focused on at Berkeley Lab, you'd be foolish to focus your efforts on this little narrow slice of cultivated microbes. You really need to cast your net wider and understand all those uncultivated organisms out there. Although I do want to note that just culturing is critical to then the next step. And so I think now that we know it's all out there, we really want to try to get more of those things in culture and study them in the lab. But the sequencing and other technologies have really opened a whole new universe. And now we can not only do 16S ribosomal RNA sequencing of just these marker genes, but we do whole genome sequencing. Everyone's familiar with this. And then we can do all kinds of downstream analyses to understand. And just as a side note, like this is a nice commentary from Tanya Wojcik and Eddie Rubin, who I work with or worked with. Eddie was a former director of the JGI, just noting that it's really technology goes hand in hand with these developments, and that it was, dating back to the microscope and then to these sequencing methodologies. And as we've evolved these new methods like single molecule sequencing and metatranscriptomes, et cetera, they continue to reveal things that we haven't been able to see before. So I mentioned kind of these extreme exotic organisms, but microbiomes are also really important in moderate environments too. Like I'm going to talk about soils today, on plants and in humans. So these are often, it's a little easier to cultivate things from moderate environments because you don't have to get extreme temperatures or pressures or things, but there's still the same situation where most of them are not readily cultivable. And so we have applied these sequence-based methods to understand soil microbiomes. In particular, I'm going to tell you about a lot of work that we've done in wetlands, which are very important for carbon cycling. And so wetlands and wetland restoration is something that I've been working on for a long time. And wetlands have amazing ecosystem benefits. They are habitat for a unique range of wildlife. They help improve water quality, but also really important to us as a Department of Energy lab, they store a lot of carbon. And the numbers here kind of depend on what you consider to be a wetland and how you quantify things, but roughly 9% of the global land area is wetlands, but they store something like a third of all the terrestrial carbon. So they're really punching above their weight in terms of carbon storage, which makes them a potential for better carbon sequestration, especially wetland restoration. And that's something people have talked about for a long time. And that's because the plants in wetlands are very productive. They take up a lot of carbon dioxide and incorporate it into their biomass. And then in these waterlogged soils in wetlands that are very anaerobic, it tends to break down very slowly. And so carbon accumulates in those soils. That is great, although there is one caveat to that, which is that those same anaerobic conditions are very favorable to the production of methane in a lot of instances. Methane is a much more potent greenhouse gas than CO2. It's at least 20 times as potent, again, kind of depending on how you do your calculations. And so that actually means that in wetlands, you could be a net carbon sink, but a net greenhouse gas source, because if even 1/20th of that carbon dioxide you take up ends up getting back out into the atmosphere as methane, you've had a net negative effect on climate change. And so our studies were really asking, well, what determines if a wetland serves as a greenhouse gas source or greenhouse gas sinks? And microbes are really critical to this because they are the ones that produce the methane. They are the ones that break down the organic matter. And so our study system is conveniently located here in the San Francisco Bay region. I don't have a pointer, but the Walnut Creek, where I was when we started this work, is just a little bit inland of San Francisco. Berkeley is just a little farther close to the Bay. And so all of these are kind of right in the general Bay region. And in fact, the San Francisco Bay Delta region used to be ringed by a lot of tidal wetlands, which are shown in green in this left-hand panel. But most of those have actually been levied off and converted to other uses over the last couple of centuries, particularly to farmland up in the Delta. These are very rich, organic rich soils that may create farmland. And in the South Bay, conversion to salt ponds has been a major use of those wetlands. And over time, people are starting to realize that that maybe isn't the best idea. And so there's been some conversion of these things back to wetlands, which I'll show in a minute. But at least in the Delta, which is where we started our studies, the effect of that conversion is that you have these freshwater wetlands, you build levees and you drain them and start farming on them, and that results in the carbon breaking down. It gets oxidized and released into the atmosphere, so it's a net carbon source to the atmosphere. And then they also tend to compact and erode. And you end up with a situation where most of these lands in the Delta are way below the surrounding water, which creates a really fragile system where if you have a levee failure, the water will come in and you end up with open water, not a wetland. And in fact, that has happened in various places. There are a few places in the Delta where there were floods that they really weren't able to recover from. And there's kind of, instead of an island, there's a little kind of hole where the water is. And they don't recover as wetlands because the vegetation can only recover in shallow water. When it's too deep, you just end up with sort of a river expansion rather than a wetland. But it was proposed at one point back in, I think it was in the 90s, that maybe a controlled flooding could help you rebuild these wetlands and both store carbon and restore elevation to those lands, recovering some of the instability there. And so we started studying this back in the early 2000s with a lot of help from our colleagues at the USGS, Mark Waldrop and Lisa Marie Wyndham-Myers. And this was a small pilot restored wetland that was created as sort of a carbon farm. It was very successful in accreting peat, these organic soils, and had a net negative greenhouse gas budget. And we went in with the really simple question of, does metagenomic data reflect the biogeochemical differences across this site, which was, it seems like a simple question now, but it was really an unknown at the time, whether metagenomic data would actually be useful in figuring out what was going on at those sites. And in fact, we did find that there were strong correlations between the functional gene repertoire of the microbial community and the variable methane fluxes across the site. And what this figure is showing here is that, I don't know, I guess I can't show the pointer here, but that in the sites that were producing more methane, there were a lot of genes for methanogenesis and related pathways, whereas in sites with less methane, there were a lot more genes for competitive pathways for anaerobic metabolism, like sulfate reduction and nitrate reduction. So this gave us confidence that we were actually learning something useful with our metagenome data. And we've expanded to studying a lot more sites in the Bay Delta. And in fact, there are a lot of wetland restoration projects out there, not all of which we studied. This is just a list from a local restoration authority listing a whole bunch of places where wetland restoration is going on. You can see it's all over the Bay Area. So we had the thought of, that salinity may be a very important factor. These wetlands that we started out studying are freshwater. Maybe we can look at both restored and historic wetlands across the Bay Delta region and better understand their carbon budgets and better understand whether restoration is having a net carbon benefit, because most of those restoration projects are not motivated by carbon at all. They're motivated by water quality, wildlife habitat, flood prevention, things like that. But we think you could have win-win and get those ecosystem benefits that those restoration projects are geared toward, and maybe you could also make them a win on the carbon front. But to do that, we really need to understand what happens below ground and whether they produce methane, which is really a big danger in a lot of these sites. So this is another image of the San Francisco Bay region, just showing the salinity gradient that exists from seawater salinity out in the ocean and the bay, all the way up to freshwater in the Delta. And my former postdoc, Susie Thoreau and Wyatt Hartman, spent a lot of time traveling around this region, going and collecting soil cores and measuring their methane emissions. And we had kind of a default assumption, which was that methane should be much higher in the freshwater sites. And that's because the sulfate in seawater should suppress methanogenesis. Sulfate reduction is known to be more energetically favorable than methanogenesis. So when you have sulfate present, that typically just outcompetes the methanogen. So we thought, we'll get high methane in the freshwater and low methane in the seawater. And to some extent, we did see that. You can see this is a log scale plot of methane, so a huge range of methane emissions from these soil cores we extracted from the wetlands. But plotted against salinity, you can see that the methane does drop quite a bit at higher salinity. So overall, that hypothesis was supported. But interestingly, in these low salinity sites, we saw really high methane production in some of the restored wetlands, which was really not what we wanted to see, because the hope for these restored wetlands was that they would be effective carbon sinks. And we also noticed that it was pretty high, not just in the freshwater sites, but in these what we call oligohaline, these ones that are just a little bit saline. And in fact, first of all, I'll say one hypothesis on this, we weren't the only ones to observe this. There were other groups studying this restored wetland, which was really more of a research site. And they said, well, maybe because it's really a very young restored wetland, the vegetation is growing really fast, it's producing a lot of labile carbon that's then getting turned around as methane. But over time, that should decrease. And I think that is true to an extent. And in fact, the Baldacci group at UC Berkeley has continued studying this wetland. It has moderated, although their estimate is that it will become a net carbon sink in 100 years or so. So it's not quite living up to the expectations of being, a carbon sequestering wetland. Though we also, so what I was about to say was that there are other studies of methane and salinity in various wetlands on the east and west coast or around the world. And our results look very similar to if you take the data from those sites and kind of overlay it on ours in similar units, similar timescales, that we see the same kind of trend and also see that high methane production in oligohaline sites. So that told us that maybe something unique is actually going on there. So we did a ton of metagenome sequencing. I know this is a super complicated slide that I won't go into all the individual genes, but we looked at genes for carbohydrate metabolism, sulfate reduction, nitrate reduction, metal reduction, methanogenesis, methane consumption. And when we looked at the genes that were correlated with methane, it wasn't as simple as just saying, more methanogens, more methane, or even more methanogens relative to methanotrophs that take up methane. But a lot of genes involved in carbon and nitrogen and phosphorus cycling were in some way correlated with methane, so that we really needed to take into account a lot of different potential pathways. And interestingly, the salinity was not the most important factor, that the methanogenesis was not, genes were not anti-correlated with salinity at all times, consistent with our thought that there's something interesting going on at that very low salinity level. Trying to build predictive models, we found that indeed methanogens and methane oxidizers were sort of the most important groups, but then also these other groups like metal reducers, and then also environmental variables were independently predictive. Things like the bulk density of the soil were actually really important in predicting methane emissions as well. And so ultimately, we kind of tried to build all of this together into models. For across all sites, it was really hard. There's just a lot of different things going on when you range from seawater to freshwater. In just the delta, where we see those freshwater and oligohaline, we saw some, like I said, the methane oxidizers and the methanogens, but also some really important effects of nitrogen cycling organisms, which seem to influence the effectiveness of those methane oxidizers. So we think those really high methane emissions result from poor uptake of methane by methane oxidizers. And that seems to potentially relate to the presence of nitrogen compounds that inhibit those methane oxidizers. Unfortunately, it means that it's hard to predict, whether a given salinity will cause a certain amount of methane. That sometimes you have low salinity with very little methane. Sometimes you have higher salinity with very high methane. And we reached out to some other collaborators who'd also looked particularly at this oligohaline region to kind of dive in more deeply to the microbiology. And my postdoc Wyatt found these other studies that had already looked at salinity manipulations. And we actually found that some people, in some wetlands, they found that adding a little bit of salinity did suppress methanogenesis. In other places, it stayed about the same. And other places, it increased methane production. And in those samples, when we went and did the sequencing, we found that it also increased the abundance of methanogens, despite this wisdom that adding seawater should always suppress methanogenesis and be unfavorable to methanogens. And in fact, this collaborator, Marcelo Ardon, had done incubations not just with artificial seawater, but with artificial seawater with and without sulfate, and found that the seawater itself had a much bigger effect on the methanogenesis than the sulfate addition. Adding sulfate didn't significantly change methanogenesis. Again, that's not, it's at a very low level. If you add a lot of sulfate, you'll definitely suppress methane, but at these low levels, it's not enough. And the salinity somehow actually boosts methanogenesis. And this has really important implications for sea level rise, because that's exactly what you're going to see as sea levels rise. Freshwater wetlands are going to become more saline, and they might actually start producing more methane in some cases. And that is not something that's part of the global predictions right now. The assumption is that increased seawater influence would actually decrease methane. So we think this has really important implications that we need to take into account. And of course, we want to just stop sea level rise, but that's a bigger problem that we're also trying to target, with Department of Energy money. So another system on the complete other end of the salinity extreme, but another one where we found that the conventional wisdom didn't really apply, was in these South Bay salt ponds. And some of you might have actually flown in over San Francisco and wondered, what is this checkerboard of funny colored things in the South Bay? And those are former salt crystallization ponds. They're no longer used for salt production. They were actually turned over to this restoration authority as part of a deal when the San Francisco airport was expanded. They decided to go remediate some other wetlands and turn them back into a more natural habitat. And so this is just showing some maps of where we've gone. This is a picture of us by these salt ponds, which the unrestored salt ponds are just this big mass of like crystallized salt and bright red microbial life. There's a ton of organisms living there, but very extreme halophiles. And so we sampled these unrestored ponds that will be restored. And I think some of them actually, since we sampled these, have started to undergo the restoration process, but the salt pond restoration project is working to restore at least 15,000 acres, and they're kind of going one at a time. And so we were able to sample these ones that really had just been left undisturbed, but hypersaline, very inhospitable to vegetation or any other life. These restored wetlands that have actually been kind of constructed to harbor wildlife. You can actually see in this image here, there's little like nesting islands that they've built for migrating birds so that they can have their nests and not have the eggs eaten by land animals. And a nearby site that does not have a history of salt production. It's not exactly pristine because it's right next to the Dumbarton Bridge, but it's something that's been a wetlands, a salt marsh for a long time. And when we started studying this, we really didn't, I didn't predict that methane would be an important part of the story at all, because again, we know that there's sulfate in all of those systems. So really none of them should have produced a lot of methane. And so this is actually all that data that I showed earlier from across the Bay Delta region. And so the expectation is, it goes down with salinity as you go up to seawater level. It should just keep going down as you have more concentrated seawater. But the actual results we got out were totally the opposite. These hypersaline sites actually produced a lot of methane, which was totally unexpected to us. And then good news, the salt marshes didn't produce methane. That was what we expected. And the restored sites actually produced a lot less methane. So this is an interesting case where nobody expected a carbon benefit from this. Again, these restorations were really geared toward things like wildlife and water quality, but it actually had a significant benefit for the methane production. This is just showing some pictures of those habitats and circling those different sites. And so one of our thoughts was, well, is there really high sulfate still in these ponds? They've been isolated from the ocean for a long time. Maybe the sulfate is depleted. It was not depleted at all. The salinity was sky high, the sulfate was sky high, and our methane fluxes were positively correlated with salinity and sulfate, exactly the opposite of the conventional wisdom on sulfate repressing methanogenesis. But after we got this result, we started digging into the literature. Has anyone seen this before? And in fact, it was one of these cases, well, yeah, back in the 80s, people had already discovered that you could get methanogenesis in hypersaline environments. And it was specifically these methanogens that use methyl-containing substrates like methanol and trimethylamine to produce methane. Those substrates aren't available to sulfate reducers, and so you can actually have sulfate reduction and methanogenesis simultaneously. And my postdoc Susie did some experiments where she added methanol and trimethylamine and was able to get methane production from these cores, whereas a more commonly known substrate for methanogenesis, acetate, didn't lead to methane production at all. So that seemed kind of like that might be the story. But we did all of our metagenome sequencing and 16S sequencing, and found that indeed those genes for archaeomethanogenesis, including methyllotrophic methanogenesis, were correlated with our methane production. But another set of genes fell out of those analyses that showed that genes for methylphosphonate degradation were also highly correlated with methane flux. And so it was, go back to the literature and learn something more. And we found that actually methylphosphonate degradation is a completely independent pathway from methanogenesis, archaeomethanogenesis, where a carbon phosphorus bond exists between a methyl group and a larger compound. and breaking that bond results in the release of methane from the compound. And that is actually, in the last decade or two, been found to be a significant source of methane production in aerobic environments, like in the ocean, that is, again, really orthogonal to this archaeal methanogenesis that we know better. And it typically happens in environments that are very low in phosphorus, because the CP bond is hard to break, but it makes that phosphorus accessible as a source for the organism to use. And so looking at the nitrogen to phosphorus ratio in these systems, and the restored, unrestored, and historic salt marsh, the nitrogen to phosphorus ratio is sky high in these unrestored salt ponds. You can see that it's like 1,000. And so that is super P limited and consistent with there being methylphosphonate degradation being a beneficial pathway for these organisms. One thing you may or may not have noticed in our earlier studies was that I didn't talk at all about genomes. And that's because at that time, with the sequencing quality and depth and algorithms available, we really couldn't assemble enough genomes to make it very useful from those delta wetlands. They were really so poorly assembled that we didn't look at the metagenome assembled genomes at all. The salt ponds are actually a lot simpler. We've been able to construct metagenome assembled genomes from them, including there's really one dominant methanogen that seems to be important in that methyllotrophic methanogenesis pathway. It was a novel, hypersaline or a halophilic methanogen we've called methanocellus. And there are actually a lot of bacteria that harbor the methylphosphonate degradation pathway, including an organism called Maravita that we've looked at in a little more detail too. But we still are, still not quite satisfied, because we just don't know, is it really just both of these things? Is it methylotrophic methanogenesis and methylphosphonate degradation, which I'm leaning towards they're both really important, but we really wish we could find out, which one of these is more important. So we're trying to dig deeper into these mechanisms of methanogenesis. We have been able to detect methylphosphonates in our system. We've done a lot of metabolite characterization with the Environmental Molecular Sciences Lab and found that things like trihalose and acetate are correlated with the methane flux. We've also found that if you add the methylphosphonates, you can get methane out of the core. So you can add the methylotrophic substrates, you can add methylphosphonate, you get methane either way. So we are working hard on incorporating all of these pathways into the metabolic models in KBase, and actually trying to reproduce what happens in these systems and see, whether we can make a better prediction of which of these is more important. And so stay tuned. Hopefully we'll get a better picture, but it does imply that overall, like, all of these pathways are really important in predicting methane. And we just, right now, I think we're still at the point where when we restore a wetland, we just kind of hope that it won't create a lot of methane. But I really am optimistic that we'll get to the point where we can better predict this and hopefully restore our wetlands in ways that better sequester carbon and prevent methane emissions. So just a couple of conclusions of basically saying the same things, but just noting both sea level rise could lead to increases in methane emissions in some estuaries, which is not part of our current predictions, and that hypersaline salt ponds may be an under-recognized methane source, because really those were not expected to be significant methane sources, but we found that they were. So with my last few minutes, I want to talk about some of the work we hope to do in the future. And so I do think wetlands are great for carbon sequestration, but there aren't that many opportunities for wetland restoration to store carbon. And we need to look more broadly for if we want to take carbon out of the atmosphere. And so this is something we're looking at in the context of the Department of Energy carbon negative shot. So this is just showing, so soil microbiomes have lots of great ecosystem benefits, but we're really focused on this carbon cycling and carbon storage. And DOE, within the last several years, has announced a number of Earth shots, which are these big short-term goals and really changing the way that we generate energy and mitigate climate change. And the carbon negative shot is one we're really excited about, that has set cost and scale and time goals for how we're going to get carbon out of the atmosphere. And one of those, there are several different pathways that are potential ways of removing carbon dioxide, but we're really interested, of course, in this soil carbon sequestration as a mechanism for taking carbon dioxide out of the atmosphere. And we were lucky enough to get the opportunity to apply for funding to do it. And we based our design on the fact that studies have found that a lot of soils, especially agricultural soils, are very below their carbon storage capacities. And so this is from one of our teammates, Margaret Torn, showing that they've determined that soils can hold a lot of carbon, especially based on their mineral content, and that agricultural soils have been systematically depleted. And other studies have found that too, that just the process of farming lands tends to lead to a breakdown of the carbon, but they could store a lot more. And so they are a prime opportunity. And then, techno-economic analyses, this is the Roads to Removal report led by Jennifer Petridge out of Livermore Lab, have looked at all of these potential carbon sequestration pathways, and they found that right now soils are our best bet. So that's showing 2025 soils are the biggest potential carbon sink, and that's with existing technologies that we could already do things that would get more carbon into the soil. Longer term, they have great hopes for things like direct air capture and bioenergy with carbon removal and storage, but those technologies have yet to be developed. And so right now, soils are a great target for getting carbon out of the atmosphere, which we need to do imminently, not, in 50 years. And so we proposed to DOE, when they had a call for energy research centers, what we called restoration of soil carbon by precision biological strategies. And the goal is the carbon negative shot. And we are targeting soil and microbial and plant factors that influence the ability of carbon to go into the soil and stay there in a stable way, because plants do fix a lot of carbon, but most of that carbon just ends up back out in the atmosphere and that plant biomass breaks down. But some of it ends up as long-term soil carbon, and that depends on a lot of physical and environmental factors that we are still learning about. So I don't have much to tell you today. We are focusing on really understanding soil carbon and its persistence. We are engineering plants to both fix more carbon and send more of that below the ground, and to cultivate microbial communities that will help convert that to long-term soil carbon storage. And so we are already doing a lot of field sampling. We are developing automated lab-based methods to better characterize the effects of different microbial and plant manipulations. We are doing deep learning to extract more information from soil metabolomes and know what really is in soil carbon, which isn't actually that well known. We're making plants that fix more carbon, and just stay tuned. And with that, acknowledge, like I said, I've had a number of really talented postdocs work on this work, and our collaborators at UC Berkeley, North Carolina State University, and of course the Department of Energy funded a lot of this work through the Early Career Research Program, and just recently this Energy EarthSat program. And then here's the JGI, which did all the sequencing that I presented today as well. So with that, I just also wanted to say, JGI, it is a user facility. It is open to everyone. So you too can do sequencing or DNA synthesis or a lot of cool genomics techniques at JGI. We have calls for proposals that are listed here. If you go to the webpage, it's really easy to find. They are open to everyone. And you don't have to be in the US. You don't have to be funded by Department of Energy. You just need to be working on problems that are important to the Department of Energy in energy and environment. So I'd be happy to talk to you. Zhang would too, I'm sure. And with that, I can take questions. Yeah.
Thank you. That was really fun.
Oh, thanks.
So you found, when you went to look for genes that were associated with unexpected methane production, you found what you expected, which was the archaeal methanogens, but you also found methane
methylphosphonates. Yeah. Yeah.
Are those encoded in the same genomes, or do you think you have two different groups of bacteria in a community? And if so, are they symbiotic in some way?
Yeah.
Or
those are completely distinct genomes, because the methanogenesis pathways are in archaeal methanogens, which are very well-defined lineage, whereas the methylphosphonate breakdown was spread across a lot of different bacterial lineages, but not the archaea. So it seems to be, I don't know, what their interactions are. And that's one of the reasons we want to do the metabolic modeling, because I don't know, I'm not, I can't think of all the possible ways that they might be handing things off, but they're all there in the same sample and working together. So.
Yeah, if they're working together, that could have a big impact on why you're seeing it there.
Yeah.
Thank you.
Yeah.
Yuri Wolf and CBI, two short questions.
Yeah.
How did you classify methanocellus? To what group of archaea?
Gosh, we do have a paper on this, and I mean, it was, I'm trying to remember what we initially gave it a different name, and then we ultimately concluded that it didn't belong in that genus because it was far enough away, but I'm not going to be able to reconstruct the name right now. Sorry. But I can send you the site.
Another tangentially
citation.
related
Yeah.
questions. Any Asgards?
Any what?
Any Asgard archaea in your samples?
I don't think they were abundant, but I'd have to look at the data to see if they showed up at
We
all.
are always looking for new ones.
Yeah. The data is all public. It's
Okay,
easy to look.
thank you.
Hi, great
Hey.
talk. So I had a question that I get a lot because I work on soil microbiomes as well. Is what can you speculate about the possible role of fungi? And I'm not sure. I'm working more with croplands and things like that, where I know fungal. It's a big thing that we're not seeing with metagenomes. Or at least the common way that we do metagenomes. So I just wanted to comment.
Yeah, I think that I agree that they're important and not sufficiently recognized in the metagenomes. And particularly, I mean, I think even when there is fungal data in the metagenomes, our pathways, our systems for assembling and annotating and analyzing tend to downplay the importance of the fungi. So yeah, in these wetlands, we have not, specifically delved into the fungi at all. In the agricultural soils, I don't think we can avoid it. We really have to because they're so important for plant interactions. And yeah, I think finding ways to look at it all together rather than trying to just kind of break apart fungi and bacteria, I think it still need to be really developed. Yeah.
Great talk. So in your earlier work where you guys were hypothesizing salinity to be important, you were manipulating salinity in the systems, and now there's this brand new pathway you think is important in looking at other things like NP ratio. Are you going to try to do similar manipulations to test whether that changes the system?
In the freshwater wetlands, meaning if we
well, actually in the hypersaline, right?
Yeah.
That's where you were seeing like phosphate now becoming something that could.
Yeah. Well, so in the salt ponds, we've determined that adding those substrates for like trimethylamine for the archaeal methylotrophic methanogens leads to methane generation, but adding methylphosphonates also leads to methane generation. So both of those can happen, but what's actually happening in the environment is a little harder. But yeah, in terms of the testing whether decreasing the salinity, et cetera, we haven't done those kind of experiments.
I guess I was more thinking because you're adding substrates that you use to like try to and seeing if it makes more
kind of force them to
Can you like add other substrates to send other pathways more important or make that pathway less kind of?
Or like to suppress the methanogenesis? Yeah, I mean, I think that that we hope to find ways, to keep it from happening. In this case, it seems like restoring the salt ponds by kind of conventional methods is, seems to mitigate the methane emissions. But yeah, this work, we're not actively working on it because we don't have any more funding on it. but I think there are a ton of questions that could still be asked for sure.
Thanks.
Yeah.
Hi. This might be entirely ridiculous, but is it even like remotely possible to create like simulated communities of wetlands, or is that just too complicated of an environment to model, like basically essentially in vitro?
Yeah, I mean, we hope to do these kinds of things. And certainly in agricultural systems, we've started working with synthetic communities, which I think is, a powerful way of understanding things. I don't think we're ever going to replicate, what's happening in soils with synthetic communities because there's just so much going on. But yeah, no, I think it would be possible, but it's not something we've actually done.
Great. Thank you.
So we do have a few more minutes, so if you have any other questions. So I'll ask one.
Okay.
I think that's a very interesting talk. I'm so glad that somebody is doing that, trying to rescue everyone from climate change, right? So I'm curious about, why farmland. So you try to think about how to implement the plan, once we found one, that there's like farmers will be motivated to adopt it.
Well, so the idea behind that, our proposed work is that, that it would be really hard to change what we're doing on like forests, right? Because we're not currently managing them, but we're managing agricultural lands all the time. And every year they get planted with something. And so if we could modify that crop to be something that better puts carbon into the soil, then, if there were the right, sort of financial incentives and it didn't affect yield, then, if farmers could get carbon credits plus the same crop yield that they we're already getting, then it would be kind of win-win for everyone, and it would just happen, versus having to set land aside for carbon sequestration, because trying to just use land to store carbon is probably not going to work. So we are focused on sorghum as our initial target, and that sorghum could be grown as an annual crop. It can also be grown as a cover crop, so that would be a good target. But to reach the scale and cost and the goals that we have, we would need to really expand it to a lot more crops. But I think corn is closely related to sorghum. So if we get something to work in sorghum, we hope it would work in corn too. And if you can make all the corn out there carbon sequestering corn, it's not going to solve climate change. We have to do a lot of other things, but it could be a net contribution to greenhouse gas mitigation, rather than currently what it is a greenhouse gas source.